Dear all, hi to all of you. Today I am here to tell you a few words about our company, Güney Agripark, who we are, where we are heading, where we are coming from, how we originated. Let me tell you a few words about history as Güney Agripark. Güney Agripark company started its activities within our previous company, Güney Agripark, and the first place was for us within Akdeniz University Technopolis. And we stayed there until 2016, and 2016, in other words, in 2016, we set up Güney Agripark and stayed within Akdeniz University Technopolis. Later on, since we needed some further area or larger amount of land to extend our activities, we moved to Aksu, Pınarlı province, to our own land to conduct our activities. And that's why we moved from Akdeniz University campus site to our own land in Aksu, Pınarlı in 2017. So what we did was, the main reason for moving those kind of activities was to extend our activities. In the university area, our total area has reached from 800 square meters to 4,000 square meters, and our annual capacity has reached to 20 million plants per year. And when the matter comes to greenhouses, in addition to our lab areas, I am proud to say that we have really very modern greenhouses, a total of 4 hectares, and it is fully equipped with all sorts of modern equipments. And we do also use the facilities of our sister company, Growfide, which is one of the best actually for growing the ordinary seedlings, and we use those 2 facilities together to get a better acclimatized conditions for tissue culture propagated plants. Talking about our physical features we have, let's move towards to human resources and what we have. I am proud to say that we have a quiet expert team, and within that expert team as human resources, there are engineers, technicians, workers with really good experience. On the other hand, when the matter comes to R&D, we are having all sorts of relatively good hardware and software to record and to conduct all our activities and keep them as a company for the future. You know, and everything is under the computer, and we are using all relatively softwares. So our young, experienced and dynamic group of researchers qualified to carry out all tissue culture studies in different product groups. One of the key features of our company that all company employees have received and will receive all necessary training on the use of to use necessary computer programs in their respective fields, and they also attend to different meetings and symposia. And as company, it is our aim to encourage all our employees to attend all relative symposia, workshop, conferences, etc. If we believe that those kind of things are really important for the education and training of our employees, no problem, we send them all and get them to receive all necessary education. By talking about those kind of issues, let me tell you a few words about our main activity area, which is plant tissue culture, or one of the area of plant biotechnology. Within the Güney Agripark company, we use tissue culture within two frames. First frame is to micropropagation studies, and second frame is to use the tissue culture studies within the breeding program. If we ask a simple question, why we are using in vitro cultures or tissue culture techniques in our company, it's possible to say that there is production throughout the year regardless of the seasons. Secondly, the generation of reproduction frequency of tissue culture propagation is too high. In a season, it is possible to obtain millions of plants from a really very small plant starting material. The third advantage of using the in vitro cultures within company, the amount of production obtained from the unit area is very high. And fourthly, since there is a clonal propagation, the plants obtained are genetically and phenotypically homogeneous. That is very good advantage. And finally, the plant materials we obtain are very healthy because they are cultured in sterile, or in other words, in aseptic conditions. For this purpose, meristem tip culture is used in our company. Why? Because the highest genetic stability is achieved in vitro clonal propagation, and secondly, viral, bacterial, and fungi pathogens can be removed from a donor plant infected with a meristem tip culture. That is how we use in vitro techniques in micropropagation. As I stated earlier, we also use tissue culture techniques within our breeding activities. And regarding the use of plant tissue culture in breeding activities, as a company, we have been using these techniques on breeding of banana and stevia. In other words, those kind of crops are very important within the frame of our activities. As a result of these activities, our company, Güney Agripark, released and registered by Ministry of Agriculture for banana varieties, namely Bonus, Pasha, Bango, and Leader. In addition to these 4 banana varieties, we also registered 3 different stevia varieties, namely Turgut 82, Levent 9, and Güney 04. So it is another main activity of our company to use in vitro techniques in plant breeding activities. Now let me tell you a few words about the plants being studied and propagated in our company. Güney Agripark has an R&D and micropropagation teams working in tissue culture for many years. The agriculture engineer in charge of the laboratory has been actively involved in tissue culture studies for more than 20 years. In other words, she has really great experience to solve all tissue culture related problems. In our company, studies on micropropagation have been carried on in more than 450 and 500 different crops. And we tested and tried to develop really best and optimized media combination for all these 500 different crops. And if we decide to name some of the crops that we are working in larger amount, I can easily say that avocado, passiflora, orchid, quite number of ornamental plants, succulents, banana, kiwi, blueberry, different kind of other berry fruits, pistachio, hazelnut, walnut, curcumin, ginger, and finally, especially some fruit rootstocks. After giving our activities on which kind of crops we are working on, of course, the natural question we are going to ask, which kind of facilities we have as company. Facilities we have is another issue that we are really proud of. In other words, our company has the infrastructure to carry out all tissue culture studies by all means. In this sense, there are autoclaves at different capacities and volumes, several pH meters, refrigerators, heated magnetic stirrers, sterile cabins, ovens, and several research microscopes in the tissue culture laboratory. At the same time, in addition to all these lab facilities, we are also proud of having very modern air-conditioned growth rooms for the propagation and rooting of plants grown in tissue culture under ideal conditions. At the stage of acclimatization of plants emerging from the tissue culture to external conditions or outside conditions, we do have really very modern greenhouses compartments with fully climate control in modern nurseries where plant growing takes place in really very much ideal conditions. Güney Agripark Plant Production Laboratory has a very modern infrastructure, and our plant tissue culture production laboratory has a total of 12 climatic chambers with an annual capacity of 20 million per year, as I stated earlier. I keep saying that we are an R&D company, and within the frame of R&D activities, we do have several collaborations. And in that sense, regarding the collaborations, we have collaborations with different research institutes, university professors, who I am one of them, and if necessary, with other companies outside when we need any kind of support. Our company has R&D collaborations within the different departments of Akdeniz University, Faculty of Agriculture, and that is one of our strong points. As I told you earlier, I am one of them, and in addition to me, two other professors from Akdeniz University are also acting as consultants within the company. So we really do have a very strong consultant group, in addition to very strong and highly educated, well-trained our own employees. One of the other issues we are really proud that Güney Agripark has been recognized, verified, and certified as an R&D center by the Ministry of Science and Industry in 2018. We are really very proud of that. And within that verified certification, we really conduct several R&D activities, and we really spend lots of money for our R&D activities. Since the major crop is going to be the kiwi, of course I am going to say a few words about micropropagation of kiwi or kiwi studies we conduct in Güney Agripark company. As stated earlier, kiwi is one of the main interests of Güney Agripark tissue culture activities, as Haver, Tomori, and Matua have been extensively micropropagated in recent years within our company. Kiwi growing places in Turkey are as follows: Northern Marmara, Yalova, Bursa, Adapazarı, and Çanakkale cities, Eastern Black Sea, Rize, Trabzon, Giresun, and Ordu cities, and Western Black Sea, where we have Zonguldak as city. These are the very important places for kiwi production. Although it is not the majority, it's a minority, there are also a few commercial orchards or let's say the plantations in the Aegean and Mediterranean regions, but they are really minor. Like in many other crops production, materials used in classical reproduction are not homogeneous, and risk of contamination with diseases and pests is quite high, where, I am repeating myself again, in classical propagation by using cuttings. For this reason, in many countries, like in our country, tissue culture has taken the place of classical propagation techniques. This tissue culture propagation techniques has many advantages over other vegetative propagation techniques. We can briefly list them as follows, which is covering also the kiwi fruit. The production of kiwi in vitro conditions is of great importance in terms of the fact that the kiwi plant is free from viruses, and that's why has a high reproduction coefficient, facilitates plant quarantine, and is free from soil diseases, pests, and other diseases, especially the varieties newly brought to our company from foreign countries. This station provides great benefits naturally. In that sense, it is possible that we can conduct international material exchange as well as in the transportation of seedlings between regions within the country. Since the meristematic cells in the growth tip tissues of the plants are virus-free in propagation with meristem culture, even if the mother plants are infected with the virus, the new plants obtained can be virus-free, as you can imagine and naturally as you may know. Tissue culture studies for plant species with economic importance in Turkey have gained momentum in recent years. The production materials used in classical reproduction are not homogeneous, as I stated earlier, and it is really there is a great risk of contamination with diseases and pests. For these reasons, seedlings propagated by tissue culture in plant propagation gradually attract the attention of all growers, including kiwi growers, kiwi farmers. However, these costly seedlings are not used by all farmers. It is thought that this problem can be solved within the developments in plant tissue culture technology. How? That's a simple question, and some answer is also very simple. We can say that plants propagated by tissue culture reduce the input cost of producers, and they bear fruit in a shorter time than those propagated with juvenile plants, and the time until the harvest time gets shorter and shorter. If we explain those kind of advantages very well to kiwi producers, kiwi farmers, I am quite sure they will be so keen on using tissue culture propagated kiwi seedlings. In Güney Agripark, kiwi propagation studies, micropropagation studies in other words, what we do? We go through a careful process starting from in vivo selection of kiwi explants until the end of acclimatization in kernels. In other words, from the field conditions where we are choosing the explant material, from the very much end where we finish the acclimatization of tissue culture propagated material. Our aim within the company is to provide the highest quality of kiwi starting materials to kiwi producers and farmers. This is exactly what we have been doing and are going to keep doing that as a promise. Since we have been keeping our promise on producing the highest quality of starting material, Güney Agripark has been also exporting tissue culture micropropagated kiwi seedlings to some European countries. Exporting kiwi seedlings propagated by tissue culture shows how good, how reliable we are. In short, I am going to say that no doubt there are many goods, but we claim as Güney Agripark we are one of the best, if not the best. In short, we say to all growers, to all farmers, choose Güney Agripark for a profitable start.